Can your brain grow new cells even as an adult? For decades, scientists thought we were stuck with the neurons we had at birth, but new evidence suggests our brains might be more adaptable than we imagined. Let's explore the science of adult neurogenesis in the next few minutes. Neurogenesis, the birth of new neurons, is a given before birth, when the human brain churns out billions of cells. Until the late 20th century, the dogma was that this process stopped in childhood. But a 1998 study in Nature Medicine shook things up. Researchers used tracer molecules, originally given to cancer patients to track tumor growth, to analyze the brains of adults aged 57 to 72 after their deaths. They found new neurons in the hippocampus, a region critical for memory and learning, suggesting humans could grow brain cells well into old age. In mice, studies show new neurons sprouting in the hippocampus and subventricular zone, tied to smell, boosting the brain's plasticity, or its ability to adapt and form memories. Fast forward to today, and the debate rages on. A 2024 study from the University of Southern California, published in The Conversation, found new neurons in the hippocampus of adults with epilepsy linked to better verbal learning, think remembering conversations. These patients, aged 20 to 50, showed less cognitive decline when more new neurons were present. Another study from the University of Illinois at Chicago, led by Dr. Orly Lazarov, detected active neurogenesis in the hippocampus of adults up to their 90s, hinting that new neurons could protect against memory loss. But here's the twist. A 2018 Nature study, led by Arturo Alvarez Builla at UC San Francisco, found no new neuron in adult human hippocampi, arguing that earlier studies used flawed preservation methods that missed the mark. So why the conflict? It comes down to method. Detecting new neurons in humans is tricky. Tracer molecules used in animal studies are too toxic for healthy people, so researchers rely on post-mortem brains. Preservation matters. A 2019 Times Nature Medicine study from Spain, led by Maria Llorens Martin, showed that brains preserved within hours of death revealed new neurons, while poorly preserved samples didn't. Lifestyle factors, like exercise, also play a role. A 2011 study in PNAS found aerobic exercise boosted hippocampal volume in humans, potentially spurring neurogenesis. But in conditions like Alzheimer's or epilepsy, neurogenesis often slows, raising hopes that boosting it could treat cognitive decline. Here's the critical insight. Adult neurogenesis in the human hippocampus is possible, with evidence pointing to new neurons supporting memory and learning, even into old age. But the process is rare, and scientists are split on how often it happens, with some arguing it's nearly undetectable. Studies showing new neurons in adults up to their 90s, or linking them to verbal learning, open doors for therapies to combat dementia or brain injuries. Yet, until we refine our methods, the full story remains elusive. This debate pushes us to rethink how our brains stay sharp lifelong. Join us for more science that reshapes our future. Subscribe and stay curious.